Hey, glad you came back. It's unbelievable that you would return for even more empowerment. That's what this is, actually. This is going to be a lecture only. There's no R associated with this. And actually, there's no points associated with it either. This is strictly and specifically to try to provide training opportunity for students in the class who are interested in data analysis and are imagining using data in the future, perhaps becoming a graduate student. Um, all these things are going to be available in the future. It's going to be this stay-at-home thing won't last forever. And some of you are thinking about being scientists. And for those of you who are thinking about being scientists or using data, this next few lectures are for you, really. And I wanted to stop and say that for the purposes of this class, really, this is a pivot point. So actually, the polynomial regression thing is a pivot point. Most statistics classes for beginners, if you're just getting started, and we started from ground zero here in this class, we assume you didn't know anything. Most of those classes would maybe get you, maybe not even to ANOVA, maybe. Now remember, I hit the hyperdrive at one point. And the reason I did that is because I wanted to be sure that you got through ANOVA and that you got a lot of experience working with actual data analysis so that you have something you can do, not just take a test, but you can actually take a data set and work with it. So I feel really good about that. And the other reason to go really fast at that point is that I wanted there to be space, and I want there to be space at the end of the class to do some of this other stuff, which is transitional. This would be a platform. This lecture, and really the polynomial lecture too, will be a platform for a whole different class, really. This is transition mode from being an undergraduate, get started in statistics and R, to, well, let's think about data analysis as a scientist and a practicing biologist. And so that's what this, so this really would be like the first lecture of a new class, maybe almost. And so a lot of the techniques I'm going to talk to you about today are things that people would actually use. A lot of these techniques you need a bunch of subsequent follow-up classes on, and we're just going to scratch the surface. But if I do so, hopefully I'll provide you with doorways and pathways for you in the future. So that you can, in the future, you can run the analysis, or you can at least be like, this is what I need. You should probably just take this video and save it so you have it. If you want to go into science, you should take all of the videos and such out of Isidore so you have it in the future in case you need to refer back to some of this stuff. So I would encourage you to do that. Okay. To the lecture. Okay, so far in this class, we've focused on analyses that are based on the testing of, testing the effect of one predictor variable on one response variable. In biology, we do studies like that quite often. Here's an example from earlier in the class: hemoglobin in different countries, and we might ask the question, is hemoglobin test a null hypothesis that hemoglobin is the same across these countries? That's just, this is the hemoglobin is the response variable. Here we just have one. Okay. Here's another one. This is from our the thing we've used a lot in the class, which is this LeBron James data set, field goal attempts and points, and there's this relationship. But we're really just thinking about the relationship between these two singular variables. Okay. We do this quite often. This is common in biology. You need to know how to do this. So we start out early in the class working on stuff like this. Or here we have, this is the fruits experiment that we do in class, but in this case, I just shared with you the old data. But again, it's just weight, and we've got these fruits. Okay, so that's a sort of one response variable divided up and these predictors. So this is common, and you need to know how to do this. It's normal in biology to have things like this. However, biological phenomenons are often multivariate in nature. That means that many different response variables must be considered simultaneously before you will understand the system and how it's working. And in a lot of people who get started in science will collect lots of data because they're excited and they're ready to go and they're raring to learn about their study system, and they will, in doing so, generate a data set like this, sometimes unintentionally. You'll have a big data set, and at the end of your study, because you're just so excited about all these different things you wanted to measure. Oh, I want to measure this and this and this and this and this. And so it's quite common for people to generate data sets that don't necessarily fit well into the sort of univariate or pairwise kind of approach we've been talking about so far. A classic example of this is a species by plot matrix. So a lot of times in biology, you'll have plots. This is plots on this axis, and these are species on this axis, and you have some measure of the abundance. You might be, this is like an ecological kind of matrix, but you might do this. In fact, a lot of people, and I'll circle back to this, but a lot of people who are working on molecular or microbial data will end up with a matrix like this as well. You have some taxa and some measure of abundance, or maybe even just presence absence in a bunch of different samples. But it's not just one thing, it's all of these. And in fact, this matrix goes this way, goes this way. There's like 80 species here. Okay, this is a lot. You can't, for example, if you wanted to compare animals or insects or plant communities between burned and unburned grasslands, you would go out into the field and you might set up plots like this, and you would measure something within those plots. And then you would want to compare them. But what happened when you got there and you started counting things, there weren't just one thing. You started counting all the beetles or all the grasshoppers or all the insects, including beetles and grasshoppers and everything else, or all the plants. Or you might sample the soil and then measure soil microbes. But then all of a sudden you've got this huge data set. And you're like, well, I just want to know whether fire changes the ecology or not. But the species are the response variable, and there's a lot of species. So then you compare them, but the data set looks like this. This is not easy to compare one to the next. Okay. So what are you going to do? What's the approach? You may also be interested in environmental parameters. This happens a lot, where the researcher goes to the field, they sample some species, and then they measure other things about the plots. They measure, for instance, on this thing, we've got pH, Maleg, this phosphorus, okay, calcium. We've got cation exchange capacity. We've got aspect, elevation, and slopes. We've got these different things about the plots that are interesting and may help explain what's going on with the response variable, which is the biology. But the biology is represented by all those species. Okay, it's not just one thing about the plot. So now you've got a matrix of species and a matrix of environmental variables. And again, a lot of people end up with this kind of situation. You can also imagine it from a microbial perspective. You're doing some sampling, but you're also measuring some characteristics of the microbial community. Just to zoom in a bit, here's plot, aspect, slope. So there's a lot of cells and there's a lot of variables going this way and this way in that matrix. Okay, so you've got 80 plots and 30 variables or something. This is a big data set, and that's just the environmental variables. Okay. So the statistical approaches we've used in class so far are not going to cut it. It's not going to work. It's not going to, these things do not yield easily to just, ANOVA or something, or t-tests. It's just can't do it. You might use those as part of what you're doing, but you can't really get to the answer just with ANOVAs and t-tests. So we need to think bigger and broader. Data of this sort are also encountered in biomedical research. Instead of plots, you can imagine some kind of microbial sampling, and the microbiome is very popular. Here I did a quick Google Scholar search. I put in the year 2000, and I got 838 results from that year. That's papers published. I put in the year 2019, and I got 39,500. This just gives you, this is a throwaway point that, or a throwaway bit of analysis to say the microbiome is really, really popular. Okay, a lot of people are doing micro or metabolome or some kind of omic kind of research. And a lot of times when you do that sort of research, certainly with microbiome, you end up with complex data sets. You can really, if you think of a kind of microbiome that has not yet been explored, you can kind of build your career around it, because scientists are really excited about how the microbial organisms drive bigger processes. And we don't know that much about them. So you can imagine, if you sample a microbial community, you're destined to come back with measurements of presence or abundance of many different organisms. You're likely to be confused about precisely what organisms those are. You may be using operational taxon unit or some other words like that. That just means it's something, but we don't know what that is. It's just some genes look different than the other things. What exactly is going on? Nobody knows. And you may also have temperature, pH, or some other things you're measuring in that gut micro environment of the gut. And so you can't simply run a t-test on this compositional information. You just can't. It's not going to work. So you're going to have to think bigger. And the goal of this lecture is to just give you notions or ideas about ways you might approach these things, doorways into which you might walk if you find yourself with such a data set. So remember early in the class, I started off talking about exploratory data analysis, and it's been something I've been bringing to your attention over and over again. And it's crucial. If you're working with big complex data sets, you're very likely to make a mistake. You're very likely to overlook important things or get lost. Multivariate data sets are a trap for scientists who have preconceived notions. You can't have a preconceived, you shouldn't have a preconceived notion. You should test hypotheses that you outline beforehand. That means you're thinking about the system. But if you're working with complex data sets, systems which you know relatively little about, then you must be open and willing to have your hypotheses disproved and having data lead you in a new direction. The quality that you present as a scientist in the very arc of your career will be influenced how well you do A and B above. So pause now and reread that. You must be open to having your hypotheses disproved, especially with a big complex data set. Even if you started your study being very excited about some idea, oh, we apply this antimicrobial soap and it reduces the microbiome by X%. Oops, we did the samples, didn't turn out that way. Turned out there was more there. So you could have a counterintuitive result. Okay, here's an example just off the top of my head. I saw this maybe a year ago where someone did a study where they had taken sanitizer wipes and wiped down toilet seats, and they found that toilet seats that were wiped more frequently had more pathogenic microbiota than those that did not get wiped down. See if you can ponder that for a minute. Now, if you were already married to the notion that those wipes made the pathogenic microbes go away, then you're just like, what? But the fact is, turned out that the wiping those things down initiated a microbiological situation that actually favored pathogenic organisms. The point is not that you can look that study up or write me an email about it later if you don't believe it. All I want to say, and the point is, you have to be ready and willing if your data point you in a direction to go in that direction, and it might disprove your hypothesis. You must be open to having the data lead you in new directions. And here's a really scary one, the scariest one of all. You have to be willing to discover that you are SOL by design. This means that your study design was incorrect to start with, and because of design itself, you cannot statistically test for the relationships that you set out to test. That's what SOL by design means. Okay? You may find yourself such. At this moment in the class, we're pivoting to the future completely. The whole class is about your future, not my future, your future. This is a moment, I've hammered this point. I'm just going to one more time and just say, you may end up SOL by design. Try not to. That's why we spent so much time focusing on experimental design, and I'm having you read literature, method section, and we look at how somebody else did data analysis and so forth, to try to get you to think about design in a good way. But you still could end up in a place where you're SOL by design, and you have to be willing to address that. It happens quite often. It is especially likely to be caused by low sample size and an inappropriate appreciation for the variation that exists in nature. The scientists did not account for the noise that's in the data set. When you have multivariate data sets, more likely that you're going to have that. Okay, you're going to have some problem in the data set where there's too much noise and you can't see anything. I've ranted about this many times in class already. So you must be willing and honest as a working scientist to discover that your data set cannot actually answer the questions that you set out to answer because the data are insufficient. Your sample size is too small. There's too much variation in the data set. It could be that once you have the data in hand, you realize that you cannot answer the question. And then once you have given it a very strong and robust run at data analysis, you have to learn from it what you can and move on. There is tremendous poison in science when a person has a data set that is insufficient, but feels that they have to get something out of it. So they start fishing for patterns so that they can say their research was productive. Please read Interleaf 8 and 9 in Whitlock and Schluter to get straight on all this. I just want to say off the top here, for any of you who are listening, maybe I'm speaking to myself because no one chose to click on this video. Could be just me in a room, which would just ranting, which is normal. But I rant whether the video is growing or not, just to say. This happens to be running here. So maybe I'm speaking to no one, but if anyone's watching this, listen, there's great poison in science. When you try to, just like, I've got to find something, I collected the data. Now, if there's a real pattern there that you weren't looking for initially, that's one thing. But if you're just fishing, okay, and making up stuff because you spent a lot of time working on it, that's no good. You need to be willing to be like, sample size. Couldn't get there, man. Okay. So how do you proceed if you're in the situation? This was just a DAO kind of just a discussion or conversation about process. So yeah, technique. Now let's talk about technique. What to do if you actually find yourself in a situation like this. Okay. First is display the compositional information. It's a light right here. Here we go. Display the compositional information. Step one. So here's an example. In this table, this data table came from plots. On the plots were a bunch of different species. Here are the species. You don't have to know what those species are. You don't have to know what these species are. But one thing you want to do is to communicate to your reader what's present in your plots. And this is one way to do it. These are summary values. Pardon me, hotline blings can only mean one thing. Okay. So these are summary values from all of the different species. So one thing you're going to want to do if you go out and you measure beetles in a prairie or whatever it is, and you've got a bunch of different species, is summarize it for your reader so your reader knows what's present. Okay. So a reader who knows these species, which I do, for example, I know what's happening in this forest now. Just scanning this. It just sets the frame for the rest of the paper. Okay. You might want to have a summary of some environmental variables like magnesium and pH and potassium, which we have in this table. So you might want to also summarize the environmental variables that you've collected, pH or what have you. You might end up with a stacked chart. I've already showed you a stacked bar chart elsewhere in the class. This is an example from a study I was involved with a doctoral student a while back, and this was a classic kind of microbiome-ish kind of study. And we got different dates across here. We've got relative abundance of all of these different microbial organisms. And it's not even close to species. This is like functional groups, okay? Firmicutes and so forth. However, someone who understands microbiology would probably get something out of this purple bar getting smaller and smaller and smaller, these proteobacteria, whereas these green ones, the firmicutes, are increasing across that time frame. That tells you something about the biology. It's a way of communicating to your reader. Okay, so now I'm going to give you a brief overview of some really important techniques. Okay, this is going to be relatively intense, just to say. However, and incomplete, but I am going to give you a start down the pathway of all of these four techniques. And two of them, multiple regression and ordination, I'm going to have separate sessions on. At least I plan to right now have separate sessions on those two. Okay. So first is I want to direct your attention to the book, chapter 18, Whitlock and Schluter, in which they discuss they have a whole section on multiple response variables. And one thing I want to say here is, just break it to you for the first time, is that there is an underlying general linear model, GLM, under regression, under all forms of ANOVA, and under multiple regression. They're like a family. They're a family of different analysis techniques. Okay. We call it the GLM, and basically understanding that helps you understand all of these different analysis practices, techniques. We can't chase it down in depth. And in fact, this would be a great pivot point, a first slide to a new class that was only about GLM and all of the different applications of it and experiments and how to run this analysis in different ways. We can't chase it down in this class, okay? We can't. But I can give you some notion of what this is all about. So we're going to walk down this table. Also, just to say, I shared with you at the start of class this basics of experimental design. Remember, I said you should read that every 30 days until you know everything in it if you're going to be a scientist. So yeah, you should do that. But the one thing I want to say is the woman who wrote this, her name is Deborah Aaron. She's a statistician at the University of Kentucky. She had GLM on her mind. GLM was the thing, and everything was a manifestation of GLM. So this packet, that's underlying all of it. And you can see it or not, but it's there. So I would encourage you to go back and review this, especially the session toward the end, where she starts talking about different models and experimental structures. Anyway, okay. Back to the top here. Okay, so what I'm going to do is, let me scroll back up, go through this table one at a time. I'm going to go all through all of these different places. And this is table 18.1 from Whitlock and Schluter, so you can review it. So we start with linear regression. So we've already done this in class. No need to talk about it here. The second one is single factor ANOVA, a completely randomized design, which is what we've been doing in class. Okay, we've done a lot of ANOVAs in class. You've proven that you can do it. I've had you do in-class assignments and also homeworks. The ANOVAs we have done in class so far have all been what we call one-way ANOVA. That means there's an underlying assumption of a completely randomized design. A completely randomized design means all experimental units have equal probability of receiving any of the treatments. Let that wash over you. It's true. Okay, but that's only the start of the ANOVA. That's only the first step in the ANOVA family. There's many other kinds of ANOVAs. For example, you might have a block design. Randomized block design is what Whitlock and Schluter are calling it. I'll put it right here. In block designs, before you apply the treatments, you set up blocks that minimize external variation within the blocks, but maximize variation among the blocks. Then you add a blocking factor into your model. That's right here. Just let this wash over you, okay? Let it wash over you. Later, if you need to run a block ANOVA, you can come back to this and then chase it down. It's not that hard, by the way. There are no imposters in this. You can do this. It's not a big deal. And you might want a block design. You can add a blocking factor to your model. You need every treatment to be randomly assigned to an experimental unit within each block. So each block has to have each treatment, and you randomly assign the treatment to the block, or you randomly assign the treatment to the experimental unit within the block. Okay. So you have to have this one, this one, this one, this one here, this one, this one, this one here. Okay. Now, an example of this would be if you were doing some sort of planting experiment and you had a moisture gradient that went from dry to wet, let's just say, or wet to dry. And that's just like the planting, like the situation you were in where you're setting up your experiment was like that, and you have to do something about it. Because if you randomly assign your treatments and there's a strong moisture gradient, you're going to get your experiments going to be screwed up, because you're going to have this known variation in the environment that could drive your response variable that is unaccounted for in the model. It's no good. So you can block that in this way and then put the block effect in the model. Got it. No problem. You might want to do it if you have a greenhouse. You might have to set up blocks because you've got a sun passing across kind of effect. So you might have a table effect that you need to block, for example. Okay. Blocking is typically analyzed as a random effect. Just let that wash over you. Okay. Or you might have a factorial experiment. Okay. Can this go right there? Don't know. Okay. You might have a factorial experiment. So like a two-way, you might have to run a two-way ANOVA where you have, in this case, this is from Whitlock and Schluter, example 18.3, where you've got herbivores plus and minus, and submersion low and high. This is like blocks on a beach kind of thing. And the study was testing how algal growth was influenced by the effects of these things. So you've got two levels of herbivores and two levels of submersion. So you've got this kind of factorial effect. This is a two-way fixed effect ANOVA is what you call that. You're assessing the surface area. By the way, excuse me, this figure is not in the book. I made that figure because I thought it'd be easier for me to explain it. The figure they use is a little bit too complicated. I want to say one thing about this. First of all, it's not that hard to do. You can do it. You can do it. You can do this. Come on, man. You can do it. Also, is that if you're going to have two-way ANOVA, interactions are really important in the model, and you got to think about it. We can't chase this down in class and pursue it, but I'm telling you, you need to read about that. It's not that easy to understand. Read about it and then use it to your advantage. That's what this is right here, interaction. You have to have an interaction effect. Let it wash over you if you don't want to do this. Another point to make. If you're going to do a factorial experiment, like we have in this case, you need to determine whether you have fixed effects or random effects. I will not chase those down in this class. I encourage you to read page 619 to get a primer in Whitlock and Schluter. But if you end up running a two-way ANOVA and you're going to have different kinds of effects, you need to determine whether or not your effects are fixed or random. And how you list them in the model is different depending on whether it's a fixed effect or a random effect. That's this mixed effect model in that table where you've got this big A and then a little b. I urge you to slow down if you encounter this. Okay. Oh, word of warning. If you have a factorial experiment and you have a lot of different levels, like this one has 1, 2, 3, 4 levels. I forgot what this was. It's from a different book. of diet or something, and then 3 levels of something else. I don't know what it was. I forgot. It doesn't matter. The full model ends up looking like this. This is the ANOVA table. This is crazy. Now you're going to have to start cutting factors out of it. It's very complicated. That doesn't mean you shouldn't analyze your data. Of course you should. But I want to encourage you to be really careful and try to set up a design that doesn't have too much complexity in it. A lot of beginning experimenters get so excited about the biology of the system they're trying to study that they create really complex models. Later, you're dividing your degrees of freedom up into smaller and smaller and smaller and smaller chunks. And you might create a model that you can't understand, that no one will ever understand. I have seen that before, where someone got so excited about all the different stuff they wanted to test, they put it all into one single experiment, and then in the end, no one on Earth could ever understand it. Try not to do that. Okay, ANCOVA. We're just walking down the steps of this table. ANCOVA is when you have a covariate. Okay, that is what you use when you have an experiment where you're applying a treatment, but you understand that there is some underlying factor, that is likely to vary in the student. No, that should say study. I'll change that. You have an a priori reason to believe this variation will influence your response variable. Thus, you measure it and include it in your analysis. So, for example, let's go back to this study. Now, what we have here is a completely randomized design. We've assigned treatments to each of these experimental units. Each square is an experimental unit. We've got 1, 2, 3, 4 treatments. So we have 3 replications of 4 treatments randomly assigned to these units. Okay, this is a standard experiment. So we can just use ANOVA on this, except what if you're measuring something about horses instead? And what if your horses that you're putting in these lots, let's just say, varied a lot in their size? You had some large ones, some small ones, some fat ones, some thin ones. You just couldn't get horses all the same. Now you're going to have some period over time where they're doing something, grazing or something, and then you're going to measure them afterwards. But you have reason to believe that the size of the horse is going to influence how they interact with your treatments. If that's true, you can measure the size of the horses first and include that as a covariate in your analysis. So you would still run ANOVA, but you would have horse size as a covariate. We will not pursue this further, but you're encouraged to read Whitlock and Schluter to follow up if you're interested. All right, there we go. So we've marched our way all the way down through this, down to multiple regression, which I'm going to talk about now, introduce at least. I do also want to stop here and say that some people you might encounter would say the first thing to do when you're going to do this, if you're heading down this GLM path, the first thing you need to do is load some packages in R and work within the context of some packages. Okay, because there's lots of packages that have been developed because a lot of people work like this. Okay, this is very common when you run ANOVAs or you're doing science. You end up in some kind of complex situation unless you're thought carefully beforehand and said, you know what, I want this to be as simple as possible. And you end up in this kind of situation. And maybe if you're even trying to be as simple as possible, you still end up needing a block or having a factorial experiment. So there's lots of different packages out there that are set up to help you do this. Now, you can code all of this just in base R the way I've taught you to. I haven't taught you how to do these particular ones, and I won't. I don't have time. But if you encounter someone who says, you must use this package, boom, thunder. You just go, all right, man, that's good. Just pick up the tools they're putting down. Just pick those up and put them in your tool case. That's the posture we assume in this class. That's you're all agreeing to that. You've agreed to it. You signed agreement earlier that you are happy to pick up whatever tool somebody puts down. That's just fine. And you'll never become a zealot. You also promised that too. I remember. So you're not going to be a zealot, but you will pick up whatever tool. So someone's like, you must install this package now. You go, oh, it's fine. It's fine. Just do it. Okay. I just want to tell you that because if you're doing this sort of two-way mixed effect ANOVA or ANCOVA or something, you're probably going to have a mentor who says, you better run this model that's in this package, or you do it this way. It's fine. Just pick up that tool and put it in your toolbox. Collect those the rest of your career. Now, multiple regression. Okay, so simple linear regression is a way of understanding the relationship between two variables, where the data analyst assumes that one variable, the predictor or independent variable, drives the second variable, which is the response, or also called dependent. Extremely useful this is, yet in most biological systems, any given response variable is likely to be determined by more than one predictor. Here's a regression where we have age and wing length of a bird. So, okay, as it gets older, the wing gets longer, and we've got this line and some variation around the line. Great. But you could imagine that nutritional status or gender or something else could be important. Okay. Here's above ground biomass in a forest and stem density. So stem density and above ground biomass. Well, maybe other things would help predict above ground biomass, not just density. What about soil nitrogen or diversity or some other thing, temperatures or something? So you might have multiple factors. Note that polynomial regression, which we did in a different section, that was, by the way, optional. So maybe you didn't do it. But so let me just say, polynomial regression is not a form of multiple regression, because you're still only working with one predictor and one response, but you're just changing the shape of the line here, right here. Just cover and then cover again, squared. That creates a quadratic effect in the line. This is not multiple regression. And by the way, another pause, polynomial regression is the first step, and what we did with drawing a quadratic curve is the first step down a pathway, right? You may have, if you're going to have curved relationships in your data set, there's other ways to do it. You might have an exponential curve. You might have a segmented regression where you've got some part that's a straight line and some part that's a curve. There's complexities. That would be a different topic. The only thing I want to say here, first of all, is drawing a quadratic curve really helped in this case. And second of all, and so it's a good skill for you to have and have exposure to. And second of all, that's not multiple regression. It's just polynomial regression, because you only have one predictor and one response. Okay, so great. So a scientist could measure a bunch of different plant species. So this is like an example that are growing in some plots. So here's a plot. We're measuring different plant species, okay? And you might want to analyze the diversity of those plants, and also measure a bunch of different things about the plots, like soil pH. In this case, the scientist would have a response variable, for example, diversity or cover of the plants, and maybe many potential predictors. You may have a matrix, that's what I've already shown you, of environmental variables. This would also apply, of course, in microbiology or many other areas of science. It might have to do with in engineering or whatever. You've got something that you're trying to predict. You've got many different predictors. You could have a slew of variables that serve as potential predictors for your response. So this is an example of all these soil characteristics. And you want to say, okay, I want to use these to predict the cover or diversity of some biological characteristic, or of some plant species, or if it's a gut microbiota, the diversity of the microorganisms in a sample. Okay, something like that. And we have all these predictors. Okay, so this is a way to think about it. So we've got a pool of variables that may each have a lot or no explanatory power with respect to some variable that we have measured. Thus, we have multiple predictor variables, and we have one response. So we have a pool of potential variables that we could use to explain the response variable. So for example, we have herbaceous cover, and we say, okay, let's explain why there's herbs in some parts of the forest and others not. What predicts this cover? We go, okay, pH is one factor of the, and we measured a bunch of stuff about the soil. So pH, so we pulled pH out of the pool up into our equation. Now we're going to build a model, and we're going to add some other things and some other things and some other things. So multiple regression is a process of figuring out statistically what suite of variables best predict a particular response. So, phosphorus. Cation exchange capacity and calcium. So we're building it like this. Now, two things to say. First is I'm going to have a whole separate session on this, including we're going to do it in R. So multiple regression, check, on the way. You can just stop here though, it's elective. So this may be the last time you ever hear about this. That's number one. Number two is that some form of this is inherent in lots of stuff in the world. Okay, this is predicting herbaceous cover based on these soil parameters, but you can predict all kinds of things. Like you might predict whether someone's going to win an election based on a bunch of different characteristics. And you can imagine that if you're going to do things like, oh, I want to forecast, you could also create simulations using models like this. And each of these parameters themselves could be a coefficient from some other equation, like when you try to predict the election or something like, oh, well, I have to have the polls. Oh, it says presidential election, so it's electoral college. And electoral college is dependent at the state level. And California has more than Ohio, for example, or than West Virginia. So I need to go state by state and predict what's going to happen in each state. And each state has its own model. And I'm going to put all those together in one big model. And then we could run simulation 1,000 times and predict it. Now, we're not doing that in this class. I'm just introducing to you the notion of multiple regression and to try to explain data that we already have. We're not projecting in this class. That would be a totally different, way beyond. RO plane has to take a right there. But we do use, I just wanted to say that this method or this technique of multiple regression is present in society in many different ways. Okay, so I have two other things to talk about. This is a long one, so you might want to pause at some point along the way, but I'm just going to keep going. I thought about splitting it up into different ones, but now I just do one. Is that right? The next is ordination. Okay, so ordination is a family of analysis techniques that uses complex information about a set of samples to organize those samples into a diagram that explains the relationships or displays the relationships. In ordination, samples are compared using some measure the scientists have made, most often as species in a plot or taxa in a sample, gut microbiota that make up a species matrix, or herbaceous plants that are collected in plots. So you have some matrix of information there's complex matrix of information about each point. Each of these would be the sample. And then you use ordination to organize those samples into a two-dimensional space. The samples are put in order based on similarity. Samples that are plotted in a two-dimensional space where the nearness of the points in the space represents how similar those samples are to one another based on the measured parameters of the samples, which is often species. Okay, so in an ordination diagram, for example, we would say these plots are more similar to each other than they are to these plots. That's the basic way you understand or interpret an ordination diagram. So you can imagine maybe these plots have maple trees in it and these plots have pine trees in it. Those are probably pine. It could be spruce. That's just a basic idea. Now they might have a bunch of other species too, because it's not just one variable. Remember, this is a whole complex community, but maybe maple dominance is what characterizes these. Yep. Yeah, good. Okay. That's it. I'm going to stop there. And I am going to say that I have a whole different lecture, a whole nother lecture on ordination, and we will chase that down, including, I hope, to have some R practice doing ordination. Okay, good. And the last thing I want to talk about in this lecture is cluster analysis. Okay, so cluster analysis is a family of techniques that uses complex information about a set of samples to organize those samples into a diagram that displays the relationship. This is exactly the same as ordination. Ordination is, let me go back up. Ordination is arraying them flat like this on points. Cluster analysis is arraying those same samples down here like this. Samples are compared using some measure the scientists have made, oftentimes a species in a plot or taxa in a sample, gut microbiota that make up a species matrix. So we've already seen this before, but this is like samples, species, some measure of abundance of the sample. This would be the thing that you use to create the ordination, or the thing that you use to create the cluster diagram. I should just pause for a second and say, analysis techniques that focus on species matrix and environmental matrix, like ordination, and there's others, but ordination and cluster analysis, that really by itself is a whole different class as well. Like that stack of things from Whitlock and Schluter, the ANOVA and ANCOVA and Block ANOVA and whatever, that GLM sort of set of techniques could take up a whole semester, like after this class. And the ordination and cluster analysis and those techniques kind of could take up their own semester too. So if this is like, feels like it's going too fast, yeah, it's going too fast. Again, all I'm trying to do is open these doors for you. So I wanted to pause here because the whole notion of a species matrix by itself and all sorts of things about the species matrix, it deserves a lot of attention, which we can't give it. I'm just introducing the notion to you. For example, a lot of species matrices are rectangular and have a lot of zeros. If you build a species matrix, look at this. This is all zeros. Why? Well, because these plots don't have that, but some other plots do. And these zeros have important implications for data analysis. I'll circle back to that maybe later, but anyway, I just wanted to say, understanding this, just even the notion of a species matrix is kind of a big deal. So you'd use a species matrix to create an ordination diagram like this, and a cluster diagram like this separately. Each plot was like a sample, and the plot is characterized by the abundance of the different species in that plot. You went to the plot, there are pine trees. You went to, you sampled the, swabbed the, lynx's paws to find the microbiota, whatever, and this firmicutes, whoa, they would show up here. So for example, okay. So each sample, this is a sample, is a tip in this diagram. That's how this works. And it's a point of the ordination diagram. The structure of the diagram is determined by the composition of the samples. So all of these samples are into this diagram, and the structure is determined by the species composition of the samples, or the other composition. You could actually put environmental variables in this too. You could put all kinds of stuff in this. This is a super flexible technique. I should also stop and say, today, now in biology, scrolling back up, ordination is really popular. For reasons I don't understand, cluster analysis hasn't been used as much. But cluster analysis is very flexible and gives you much the same information. And actually, in a way, is more satisfying to look at, actually, to me. It's not so smeary, it's more clean. Yep, okay. So cluster analysis is a family of techniques that uses complex information about a set of samples to organize those samples into a diagram that displays the relationships. As in ordination, the samples more close to one another are more similar, points that are farther apart are more different. These 2 points are more similar to each other than either of them is to this one. The structure of the diagram is determined by the composition of the samples. To oversimplify for explanation, a lot of pine trees could be on one side and a lot of maple trees could be on the other. You can see this divide here, so I'm going to show you again. One of the many reasons someone might want to use ordination and also cluster analysis, many of the reasons someone might want to use ordination are also reasons to use cluster analysis. I just said that a second ago. The point is, this is a super interesting technique. And if you end up working with multivariate data, you should probably look into this. It can be better than doing an ordination, even though ordinations are more popular. Statistically, it's just as valid. What if you're interested in understanding how similar or different samples are? You can use a cluster diagram to demonstrate that. And cluster analysis will provide you with a statistical estimate of the strength of the pattern. One useful attribute of cluster analysis is you can see the relationships expressed in these brackets. For example, in this diagram, you can see these samples sort of hold together here. This one seems to be by itself. This is unique. So if I was studying this system and these were my samples, I go, these hang together, these are really tight here. Something weird about S86. Got to look back into that. Did somebody mess something up? Are the data good? Did something happen there? What is that? So that tells you something about the samples. And we can see that all of these are broadly dissimilar than all of these. These are divided up. There's a big divide right here in the data set. This can be something super interesting. For example, if you were doing, for example, if your study was about the effects of fire on the communities of these grasslands, and your plots arrayed themselves this way in your cluster diagram, you'd be happy about that. Say, man, this is really cool. I can really see, I have good statistical evidence that the fire is impacting the community, the whole community. The structure of this thing is created by all of the species in the plots. They all get to be part of it. So you're looking at holistically in this way. Okay, so here's some things you need to know. First is that things you need to know. Oh wait, so we will not run cluster analysis in R in this class, at least so far. If I come up with it, I will, but I don't think so. I think we're going to run out of time. But it's not particularly difficult, and you can do it. If you've made it this far in the class, you definitely can do cluster analysis. If you ended up wanting to, and you have the appropriate data set, you can do this. There's no imposters here. Okay. Things you need to know that are really simple things. One is that you need to select a distance measure. In this case, I'm using Sorensen's distance. That's a sort of famous one. Again, this is this cluster analysis should take a month, okay, of classes to really understand it. But I want to get you I just want to give you introduction. There's many others. Sorensen's distance is kind of a popular one. But you need to do some reading to figure out which one you're going to use. The second thing you need to know is that that cluster analysis has a clustering technique as well. So in this case, I use what's called UPGMA clustering. That's, again, a popular one, common. There are many different styles, and you need to do some reading to figure out which one you want to use. You have a distance measure, and you have a clustering technique. Okay. Okay, so here's data sets that are analyzed, the same data set. So these samples are all the same, S83, S84, and so forth. So the information in the data set is the same, is identical. What's different is the distance measure and the technique. This is UPGMA and Sorensen. This is correlation and farthest neighbor. Don't worry about it. Let it wash over you. Those are just techniques. You can see this broadly similar, but not identical. The length of this is a little bit different. We're still having 2, 3. It's pretty similar, although this one, remember, 86 didn't show up there. It showed up somewhere else. So maybe it's right there. I can't tell. So it's not the same exactly. So this matters. And if you're going to do cluster analysis, you need to read about these and figure out why you'd want to use them and make a good choice. Here are two that are using the same clustering technique, but different distance measures. So this is correlation and farthest neighbor. This is Sorensen and farthest neighbor. Let's take a look. They're quite similar, but not totally identical. Yeah, this fork right here is interesting. So again, you have to figure it out and think about what exactly do you want to pursue, which of these techniques is best. It is worth saying that when you look at those two, remember the patterns I showed you before, which has this major fork in the data set, are present in all of them. So the big pattern is there, but some of the details are not the same. The main idea right here is that you're going to want to explore, if you want to explore cluster analysis, you're going to have to learn about which technique do you want to use and which distance measure. Okay, this concludes my overview of multivariate data. I hope you enjoyed it, and we'll see you next time.